Hello, my name is Brian Chen, and I'm a professor at McGill University at the Center for Research in Neuroscience. And today, I'll be talking about experience-dependent synaptic and anatomical plasticity. So what is experience-dependent synaptic plasticity? Well, it's basically the changes within the brain that occurs within our nerve cells or neurons after we experience something new. So plasticity is the ability of a nerve cell or a neuron or any type of object to change. And it is the synapses or the connections between these nerve cells that we think are changing after we experience something new. So for example, let's say we experience or learn or remember something from the outside world. We assume this is making some type of change in the brain, whether this be a change in a single nerve cell or neuron shown here, or at the level of a single synapse, the connections between neurons, at the level of molecules here, or the activity or the anatomy or the structure of these nerve cells that are changing. So the series of events that occur during experience-dependent synaptic plasticity are basically some type of sensory or motor experience from the outside world. That, of course, causes neuronal activity within our brains or within the brain, changes within the synapse, which leads to some type of long-lasting functional circuit modification. And for this next series of talks, I'll be focusing on the experimental evidence that has been described at the level of synaptic changes and anatomical changes that leads to a functional circuit change. So again, at the level of molecules, we could think of it as molecules, synapses, and circuits. So to be clear, of course, this type of question has been examined for many thousands of years, dating back even in the time of the ancient Greeks, for example, where Plato wrote about it in Meno almost 2,500 years ago, where he postulates, for example, can you tell me, Socrates, whether virtue is acquired by teaching or by practice, or if neither by teaching nor practice, then whether it comes to man by nature or what other way? So in this case, he used the example of virtue, but this can be anything, and this is also classically described as the nature versus nurture argument, which of course we now know is not a very accurate reflection of the question, but still, is it that all of the things that we experience in life just come straight from nature, hardwired within our brains, so that we never have to learn anything? We of course know that's not true. We also know that there are some things that are innate within us, the ability to breathe as soon as we're born, for example, and so it is clear that some neural circuits require experience to refine themselves so that we can learn how to ride a bicycle, for example, and get better and better at things. Or some neuronal circuits are completely hardwired where they're able to function as soon as the animal is born. So the experimental evidence for experience-dependent plasticity came from the groundbreaking work of Torsten Wiesel and David Hubel, who won the Nobel Prize in 1981 for their groundbreaking work in information processing of the neocortex, or specifically the visual cortex, the part of the brain that deals with visual information. So this paper was the first in a series where they showed that deprivation of sensory experience, for example, the deprivation of one eye for light, caused profound changes within the brain of the cat. And so here are the introductions to their Nobel lectures delivered December 8, 1981 by Torsten Wiesel and David Hubel. And I'll just read quickly here the introduction to Torsten Wiesel's Nobel lecture. In the early 60s, having begun to describe the physiology of cells in the adult cat visual cortex, David Hubel and I decided to investigate how the highly specific response properties of cortical cells emerged during postnatal development. We were also interested in examining the role of visual experience in normal development, a question raised and discussed by philosophers since the time of Descartes. I will first start with a brief overview of the visual system. So here we're looking at the human brain. Here's the left eye and the right eye, and it's looking at this picture here. The visual field can be separated, let's say, for example, in the left and the right side in blue and in red, or from numbers going from left to right, 1, 2, 3, 4, 5, 6, 7, 8, 9. And here is the circuitry, the neuronal circuitry within the visual circuit that I'll go through that dictates how information is processed from light into activity within the brain. So here we have, for example, on the left eye, you'll notice there are a series of red lines that are shown as wires. These will be bundles of axons that come from the retina, a thin sheet of neurons on the back of the eye here, that then synapse onto a structure within the thalamus of the brain called the lateral geniculate nucleus, or the lateral geniculate body, LGN. And that's the first synapse that they take along the way. And then after that, these neurons here send their axons into the primary visual cortex here, also called the striate cortex. What you'll also notice is that the axons, the wires that come outside of the eye, can be designated in two different colors, red versus blue, for clarity. And here you'll notice that a lot of the axons closer to the nose side or the nasal side cross the midline of the brain here, so that all of the axons from on the blue side that take input from the left visual field cross into the right hemisphere of the brain. The same is true for the right eye, the right side, which takes in information from the right visual field, crosses, so that all the information that's coming from the right visual field, whether it's coming from the left eye or the right eye, let's take a look at the numbers. The red side here, 6, 7, 8, 9, are then all processed here in the thalamus, or the lateral geniculate nucleus of the thalamus, as well as in the striate cortex, in the visual cortex here. And the same is true here. And so this is called the ipsilateral projections, ipsilateral meaning same side, or contralateral projections, contralateral meaning opposite side. What you'll also notice is that the organization of the visual circuitry is topographic, meaning the information is preserved from left to right in the visual field, and this is also preserved anatomically within the thalamus as well as within the visual cortex, so that there is a steady and smooth arrangement of the visual field within the thalamus and the cortex, so that neurons within the thalamus and neurons within the visual cortex can correspond to specific areas within the visual field. The area in stimulus space that a neuron responds to is called a receptive field. And this is an important concept in neuroscience, because this area in stimulus space can be, for example, for a visual neuron, that area within space in vision that the neuron responds best to, as well as, for example, an auditory neuron, the area within this stimulus space in frequency that an auditory neuron responds to, or, for example, a mechanical sensitive neuron, an area within the skin that a neuron responds best to, which could be over here, for example, or on the other side of the body. This stimulus space can also refer to the area that the neuron responds best to in terms of the inputs that it receives from other neurons, where it can integrate all the information coming from it from other neurons, and this can also be defined as its receptive field. The way that Hubel and Wiesel could measure these receptive fields was using a technique called microelectrode recordings, which allowed for the measurement of electrical signals of neurons, the language of neurons, as small as microvolts or millionths of a volt. For example, by placing the electrode in the brain of a cat attached to an amplifier and oscilloscope, they can then measure out the electrical recordings within single neurons within the brain as they presented visual stimulus to a cat. So, for example, if they were to measure the receptive field of a single neuron in the visual cortex of the cat, they might project the stimulus onto a screen in front of the eyes of the cat, and then you can now determine different types of receptive field properties of this neuron. So, for example, you can look at the size of the receptive field, whether it's a circle or a small circle or big circle, a ring, annulus, for example, here, or a line or moving lines and moving bars. So using this technique, Hubel and Wiesel discovered an important property of neocortical neurons within the brain of mammals, that is, how they can process information from more simple information to more complicated information, integrating the information from upstream neurons into downstream neurons. So let's take, for example, what would happen if we shined light within the receptive field of these visual cortex neurons upstream of this integrating neuron. If we shined light here directly within its receptive field, then of course this would cause this neuron to spike. And if we also shined light here, this would cause this neuron to spike here. And the same goes for these. It's important to note also that these neurons have a classic center-surround receptive field in the sense that a light stimulus activates at best when it's this size here and is inhibited by a light stimulus here, which is designated by these minus signs. And so a light stimulus that is this exact size here would cancel each other out because it would not only stimulate this area here, but also negate this area here, which would cause it to fire less frequently than, of course, if the light was shown just within this area here. So if we shine light here, 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 and there, you'll see a series of action potentials or spikes generated by these neurons that signal that it received the proper stimulus in stimulus space. If we shine a bar of light, of course, this will generate the same signals from this neuron, and this downstream integrating neuron will, of course, generate a much larger series of action potentials as it is now integrated the information into a more complicated receptive field. And it is this discovery by Hubel and Wiesel that neurons could integrate information from more simple receptive fields to more complicated receptive fields to more complex to hypercomplex receptive fields. This property allows visual cortex neurons to also be selective for different orientations of a light bar. So, for example, they projected a light bar of various orientations onto this screen and then recorded the response properties, or the spiking output, the action potential output of these visual cortex neurons as a function of different orientations of these bars of light. What you'll notice is that this stimulus in this orientation produced only a few action potentials or spikes. This orientation produced a few more, whereas this 90-degree orientation produced lots of action potentials for this neuron within this animal. And this orientation produced a little bit fewer. This one and this one, of course, produced very little to zero spikes. And this is summarized here on the right-hand graph. On the y-axis, we have the cell's response. And on the x-axis, we have the orientation of the bar, and you could see a tuning curve, or the receptive field properties of this single neuron and its response to these different orientations, and it likes a 90-degree up and down bar the best. And as they continue to record for more neurons within the visual cortex, Hubel and Wiesel made another very important discovery, which is the columnar processing of the cortex. And this was another important discovery because for many hundreds of years, neuroscientists and anatomists thought that the information processing of the brain was through the different layers of the cortex. That is, as you go deeper within cortex, you would encounter different types of cells and layers of different thicknesses with different cell types and shapes. But what they discovered was as they inserted the microelectrodes within the brain along a column deeper into the brain, all of the response properties of these neurons stayed the same. They were always tuned best to a specific orientation of a light bar, which is shown here. For this electrode penetration 1, 2, 3, 4, 5, and 6, you'll notice that all of the neurons respond best to this single orientation. Whereas, if we look at the brain from the face down, you could see that of these 32 recordings performed by Hubel and Wiesel in 1963, they have lots of different orientation selectivities as you go from 90 degrees all the way to 0 and 180 degrees, and all of the smooth transitions in between. We can now use more modern techniques using optical imaging, where we can make a map of the entire brain, looking down again on the brain, and overlay, using a color code, all the different selectivities or preferences for the different orientations of an entire region of the brain. So this scale bar here, for example, is 1 millimeter across the brain, where we're looking at now millions of neurons and the response properties. And so you can see the smooth transitions between orientation selectivity for 0 degrees all the way to 90 degrees and everything in between. So the receptive fields of many neurons can be measured, and maps can be made of the receptive fields or the response properties of many different cells or neurons can be cataloged and analyzed, and it is using these methods that Hubel and Wiesel determined what is changing within the brain as the sensory experience of the animal is altered. And I will now go through the classic experiments that Hubel and Wiesel did to determine these specific properties that are changing within the brain as they altered the sensory experience, the visual experience of the animal. Again, we have the schematic of the visual system of the mammalian brain, where we have the left and right eyes, and the axons from the retinal ganglion cells project into the thalamus, into the lateral geniculate nucleus, and then from there, neurons within the lateral geniculate nucleus send their axons into the visual cortex, primary visual cortex area V1. And so then you can do recordings from the cortex or from the thalamus. And using this technique, Hubel and Wiesel measured the response of different neurons within the lateral geniculate nucleus or the cortex to stimulation of either the left or the right eye, which could then be determined as being the ipsilateral or the contralateral eye. In other words, if they were recording from a cortical neuron in the left hemisphere, by stimulating the left eye, that would be an ipsilateral response. And then by stimulating the right eye, that would be determined as a contralateral response. And so they generated ocular dominance histograms, where on the y-axis we have the number of cells that responded to light stimulation of either the contralateral or the ipsilateral eye, or to both eyes equally, as well as different measurements in between. So, for example, if they were measuring from a neuron in the cortex in the left hemisphere, then responses from predominantly only the left eye would be determined as monocular only and be given a score of a 7. So that one cell would count as 1 here. If they recorded from another one in the same hemisphere and it responded predominantly to just the right eye, which would again be contralateral monocular, it would be given a 1. If it gave weak responses to, a little bit of weak responses to the right eye, and then a little bit stronger responses to the left eye, it might fall into this category. If it gave spiking responses to both eyes equally, then it would be categorized as a binocular cell. So by using ocular dominance histograms, they could determine the distribution of different neurons within the cortex and how they responded, whether they responded best to contralateral visual stimulation, ipsilateral visual stimulation, or as we can see, mostly to binocular stimulation. And they can even, of course, also segregate these ocular dominance histograms to just neurons within specific layers of the cortex, for example, layers above layer 4, 1, 2, and 3, or within layer 4 only, or lower than layer 4, 5, and 6, for example, or restrict this ocular dominance histogram to just the neurons that they had recorded from the thalamus. To understand how changing the visual experience of the animal might shift these ocular dominance histograms, Hubel and Wiesel then took the same age kitten, or a 3 to 4 week old kitten, and then sutured one of the eyelids shut for a period of 1 week. After the 1 week was over, they released the suture and allowed the animal to experience normal vision again, and then recorded again from populations of visual cortex neurons and generated ocular dominance histograms. And what they found, as you can clearly see here, is that the distribution of cells that responded to the one eye that remained open completely shifted, so that the majority of neurons they were recording from within the cortex responded best to the open eye, with a small minority responding to both eyes or to the closed eye. This was an important and groundbreaking experiment because they simply altered the sensory experience of the animal. They didn't damage the eye, they didn't damage the neurons within the eye or any of the neurons within the downstream neurons within the thalamus or within the cortex. And by simply just depriving the animal of visual experience for about 1 week, they found that the vast majority of neurons within the cortex were then now responding to the non-deprived or spared eye. So they next wanted to determine what would happen if they sutured both eyelids shut. And so they did the same experiment, but this time by suturing both eyelids shut for a period of 1 week, opening them again, and then recording the responses from one eye versus the other eye, they found that the ocular dominance histogram was somewhat restored in the sense that they now responded equally best mostly to both eyes, with some cells responding best to just stimulation of the contralateral eye or the ipsilateral eye. So they were surprised by this finding, of course, because, as they put it, it was as if the expected ill effects from closing one eye had been averted by closing the other. To be clear, of course, this binocular deprivation caused the cats to be functionally blind. When they matured into adult cats, they could not see at all. And from this, despite the fact that the ocular dominance histogram was restored, Hubel and Wiesel concluded that binocular deprivation restores the balance in the active inputs to the cortical neurons by probably weakening all the visual activity equally. By recording from lateral nucleus neurons, they also found that the binocular deprivation did not affect these neurons, but it's important to note that it was difficult for them to obtain large numbers of these recordings because the visual responses from these cortical neurons were sluggish and unreliable. And then further studies showed that loss of visual experience causes a developmental delay which affects the plasticity of the more complex visual processing or downstream of primary visual cortex, which then explained why the adult cats were essentially blind. In the next series of elegant experiments, Hubel and Wiesel then wanted to determine the mechanism by which these shifts in ocular dominance histograms could occur. So what they did was they cut the lateral rectus muscle here, which controls the movement of the eyeball. And in doing so, because the medial rectus muscle is spared, this causes an artificial squint in the kitten as the eyeball is pulled toward the nasal direction. This experiment is critical because they wanted to test whether simply changing the pattern of activity rather than the amount of activity that occurs throughout the visual system coming in from the left and right eyes would change the ocular dominance histograms. And what they found was in cortical neurons, they all became predominantly monocular, as you can see here from this ocular dominance histogram. So almost all the cells responded primarily only to the contralateral or the ipsilateral eye, with very few binocular cells. If we compare it to the control case, you will remember that the distribution of cortical cells was an upside-down U shape with predominantly binocular cells in the cortex, whereas now, after induction of the artificial squint, or strabismus as it's called, this causes almost all the neurons to become monocular. And this led to the competition hypothesis. As they state, if one eye is not stimulated, the fate of its projections in the central visual pathway may partly depend on whether or not the other eye is stimulated. In other words, what they can conclude from these experiments is that the inputs coming into the cortex directly compete for cortical territory, and that patterned correlated activity is required. And this is because the two eyes will never see the same visual space. And that is because one of them is controlled normally while the other one will always have a bias towards another direction. Because they never have the same pattern of correlated activity, then the binocularity is lost because the activity between the inputs will no longer be correlated. So in summary of the electrophysiological experiments of Hubel and Wiesel, by depriving one eye of sensory experience for 1 week in the juvenile cat, this shifts the receptive fields of the cortical neurons to the spared eye. Binocular deprivation, on the other hand, restores this competition between the two of them, but delays development. And then the induction of the artificial strabismus abolishes binocularity within the cortex. And then further studies established that the correlated activity between the two eyes in strabismus is abolished in visual circuits downstream of primary visual cortex. In another groundbreaking experiment, Hubel and Wiesel wanted to determine when within development changing the sensory experience of the animal would change the responses of the neurons from the cortex. And so what they did was, of course, to suture the eye at different times in the animal's life and then determine what would happen at adulthood when they were recorded from the cortical neurons. What they found was that monocular deprivation of the cat only shifted the response properties of cortical neurons if it was performed within a critical period or time window during the development of the cat. In other words, if you monocularly deprive the adult cat and then measure the responses within the cortex, they would still be normal and have a normal binocular distribution. That's shown here. Again, in a normal kitten, where the y-axis we have the number of cells that respond to either the contralateral eye, the ipsilateral eye, both eyes equally, you again have this upside down U shape. If you monocularly deprive the kitten for a period of about 1 week, then the responses of the cells within the cortex shift towards the spared eye, with very few of these cells responding to the deprived eye. If you monocularly deprive an adult cat for even a period of up to 1 year, after removing the sutures and recording from the cortical neurons, you'll see that the ocular dominance histogram remains predominantly binocular, just like a normal cat. And so this was an important finding because although it had been known for some time that early brain development or development of any organ in the animal has a sensitive time period where the experience of the animal will have a profound effect. However, Hubel and Wiesel had discovered the critical period, which is a fixed developmental time window where proper experience is absolutely required for normal adult function. And changes that occur within neural circuits during this critical period is irreversible. As the window closes, quote, either connections once lost are incapable of properly reestablishing themselves, or the failure of recovery may simply be a matter of age. And of course, the same experiences in the adult will not induce synaptic plasticity. To determine the anatomical changes within the brain, that is, how the structures of neurons change after visual experience-dependent plasticity, Hubel and Wiesel then turned to a technique called radioactive tracing. And what they did was inject a radioactive tracer within one of the eyes, and this is then taken up by the cells of the eye and transferred across the synapse and across every synapse along the visual circuitry in the system. And so as it goes from the left eye, for example, to the thalamus into the visual cortex, they can then dissect the visual cortex, which we're looking at here. This scale bar here is 1 millimeter. And if you place this up against a substance that they used a long time ago called film, the radioactivity would then produce a signal, which you can now see as these white stripes. And now you could see also why the visual cortex was often called the striate cortex, as the signal from the radioactive tracer from one of the eyes then creates these clear striped patterns which labels its synaptic territory within the cortex. If they did the same experiment, however, on a monocularly deprived kitten, shown here, and injected the radioactive tracer within the spared eye, you can clearly see the radioactivity signal on the film where you see an increase in the territory that's taken up by the radioactive tracer from the spared eye. The images produced by Hubel and Wiesel were bulk images of many neurons that had taken up the radioactive tracer that created the radioactivity signal on the film. Later studies showed at the single cell level that the axonal arbors coming from the thalamus, as they terminated within the cortex, coming from, for example, the spared eye in a monocularly deprived kitten, were bushier or had denser arbors compared to the deprived eye, as they were less dense and had sparse axonal arbors. And this was even the case for kittens that had monocular deprivation for only 1 week. So to conclude Hubel and Wiesel's landmark experiments that added to our fundamental knowledge of how the brain works, they discovered how neurons tuned to specific features are organized from more simple neurons to more complicated neurons that integrate this information. They also characterized how information processing flows within the cortex in a columnar fashion, and how experience influences the specific properties of this neural circuitry, as well as the mechanisms by which these neural circuits are refined, and they discovered the critical period for experience-dependent plasticity. Critical periods for other stimulus features have now been characterized in further studies. For example, ocular dominance columns in different species and model organisms, the orientation selectivity for visual cortex neurons, the whisker to barrel map formation in the rodent cortex, which I will talk about further, or for example, tonotopic map in the auditory cortex or absolute pitch within humans, and human language. So now I will discuss experience-dependent plasticity and the critical period for whisker receptive field tuning in rodents. Now I'm going to talk about a model system that is near and dear to my heart called the rodent barrel cortex. I used this model system for my entire PhD thesis. The rodent barrel cortex is an area within the primary somatosensory cortex, or S1 cortex, of rodents, such as rats and mice, that corresponds to the whiskers of the rats or mice. So here we're looking at the mystacule vibrissae, or the large whiskers on the snout of a rodent. And you can see there is a one-to-one topographic organization from whisker to barrel. So for example, this whisker here is the A1 whisker, which corresponds to the A1 barrel. Here we're looking down on the cortex, or on the surface of the brain, at the primary somatosensory cortex, where this is anterior, this direction is posterior, this would be medial, and this is lateral. One advantage of using the rodent barrel cortex is its history of use in experience-dependent plasticity, where one can simply trim one of these whiskers to change the sensory experience of the animal, and then we know exactly where to look for any of these changes in experience-dependent synaptic plasticity to occur. The rodent barrel cortex is organized just like any other canonical cortical circuit in a columnar fashion, where a single barrel will correspond to a single cortical column. It is called the barrel cortex because when you look at a cross-section through the cortex, shown here, layer 1, layers 2 and 3, layer 4, layer 5, and layer 6, the dense innervation that comes from the thalamus axons that terminate within layer 4 form these barrels. The development of the rodent barrel cortex mirrors the development of most other sensory cortical circuits, where the onset of the behavior, for example whisking, corresponds to the wiring and the development of the cortical circuitry. So, for example, whisking behavior within rodents starts to occur at around postnatal age 10 days and increases till around postnatal age 17. And this corresponds to a large increase in the synaptic density within the barrel cortex. So, to measure the response properties of neurons within the rodent barrel cortex, we can simply use electrophysiological recordings of these neurons, and then by stimulating single whiskers on the snout of the rodent, we can then map out the receptive fields. The receptive fields for these single neurons are organized in a stereotypical center-surround fashion because of this topographic or somatotopic organization, where stimulation of the corresponding principal whisker of a neuron within the barrel cortex elicits the strongest response, and then stimulation of the first-order surround neighboring whiskers elicits a less strong response. Using these electrophysiological techniques to record from neurons within the cortex, the first person to demonstrate synaptic plasticity within the rodent barrel cortex after changes in the sensory experience or the whisker experience of the animal was Kevin Fox in 1992. And what he did was to clip all of the whiskers except for the D1 whisker. That is, he would perform a sensory deprivation, removing all of the whiskers except this single whisker here, the D1 whisker, starting from birth all the way up until about 30 to 90 days, so for about 1 to 3 months. But before I get into the experimental results, let's take a look at the control condition, where at around 1 to 3 months of age, if you record and map the receptive fields of neurons within the barrel cortex, let's say neurons from the layers of the neocortex 2 and 3, or the layers 4, on the y-axis we have, again, a histogram, or percentage of cells, here from 0 to 70% of cells, that respond best to the principal whisker, or stimulation of the principal whisker, versus the D1 whisker. And so the principal whisker in this case might be, for example, the D2 whisker, or the C1 or E1 whiskers. And then the first surround whisker, neighboring whisker, would be the D1. And what you can see in the control case is that the majority of neurons respond best to its principal whisker. That is, if he's recording from the D2 barrel, the majority of the neurons respond best to the D2 whisker. And then these small tenths of responses here are just the normalization between the principal whisker and the D1 whisker, from 1/10, 2/10, 3/10, 4/10, all the way to a full response of the D1 whisker, of which no neurons responded best to the first order surround whisker, the D1 whisker, in layers 2, 3, and layers 4 of the cortex. Now let's see what happens after many months of deprivation while sparing the single D1 whisker. What you'll notice in both layers 2, 3, and layer 4 is that many, many more neurons respond now to the D1 whisker, the spared whisker, even though the principal whisker, anatomically defined, is the D2, for example, or the C1 whisker, in this case, the P. And this is highlighted here in the actual paper. Note that the D1 vibrissa rarely produces a greater response than the principal vibrissa in layers 2, 3, and 4 in the control case. But after the deprivation and sparing of only the D1 whisker, essentially the dominant vibrissa has changed for these cells from the one that would be predicted by their anatomical definition or their anatomical location to only the spared whisker, D1. Evidence for a critical period for this experience-dependent synaptic plasticity within the rodent barrel cortex came from the lab of Karl Svoboda in 2001, where I performed my PhD thesis. Again, if we focus our attention to the top part of the slide, the control conditions where the animal has all the whiskers spared, you'll see for an animal at postnatal day 14 in the receptive field map for any of these neurons at postnatal day 14, it has a stereotypical center-surround organization where stimulation of its principal whisker, here principal column 0 and principal row 0, elicits the strongest response, for example, several tens of millivolts, and then of course has less strong responses to the first order surround whiskers, for example, one column over here or one column away, or second order surround whiskers here, 2 rows away or 2 rows away here. And the way to read these maps, whether it's at postnatal day 14 or 20, you can see is that each one of these delineations is about 0.6 standard deviations or 1.2 standard deviations, and so on. Next, what they did was to deprive the animal of all the whiskers by clipping them at specific ages, specific time windows to determine when and whether there was a critical period. So, for example, if you were to deprive an animal at P12 for 2 days or P14 for 2 days, then after you let the whiskers grow back to again map the receptive fields of neurons within the barrel cortex, you'll see that the receptive field map in the deprived case, when the animal has all the whiskers clipped at postnatal day 14, produces an abnormal map where stimulation of the principal whisker sometimes does not even produce the strongest response. In some cases, for example, it can be produced by the first order or even second order surround whiskers. Whereas if this full field deprivation was performed at later ages, for example at postnatal day 20, the receptive field map produces the similar control condition center surround organization. What are the anatomical correlates for this synaptic plasticity? In the next series of experiments, I will show you how by simply observing the structure of neurons, this can allow us fundamental insights into the mechanisms of how the brain changes in response to changes in the sensory experience of the animal. The way we perform these experiments is by using a technique called two-photon laser scanning microscopy. And what this essentially allows for is by using a laser that produces very high fluxes, this can allow high-resolution imaging of neurons deep within the brain of the living animal. The way we image these neurons at high resolution is by using a molecule called green fluorescent protein, or GFP. And this molecule is expressed or fills the entire cell or neuron in only a small number of these neurons within the rodent barrel cortex. So again, we're looking down onto the brain of the living animal, and you can see there are 3 neurons called pyramidal neurons in this field of view. This scale bar here is 100 micrometers or 100 microns, and here's a side view of one of these pyramidal cells in vivo in the living animal. The structures that we focus on to correlate these changes in synaptic plasticity are the sites of synaptic contact within the brain called dendritic spines. And here's a cross-section through a dendritic spine in this electron micrograph that was produced using a technique called electron microscopy. These dendritic spines are very small. As you can see, the scale bar here is 0.25 micrometers, or 1/4 of a micron. And this is the synaptic cleft with the presynaptic axon, synaptic vesicles, and these tiny appendages that protrude from the dendrite of a nerve cell. The reason why we focus on dendritic spines is because they are the structural correlates of synapses, where more than 90% of excitatory synapses occur on dendritic spines, and almost every single dendritic spine has a synapse. So what happens when we image these neurons at high resolution in the brains of the living animal?
This movie will show the dynamics of dendrites and axons of neurons in the brain of a living rat imaged every minute for 2 hours. These neurons are pyramidal neurons in layers 2 and 3 of the somatosensory barrel cortex in a 10-day-old rat. The neurons are imaged in the living animal using 2-photon laser scanning microscopy and are filled with a fluorescent molecule called green fluorescent protein, which allows the structure of just these few neurons in the field of view to be observed, surrounded by a dark background. The scale bar in the upper left corner is 5 micrometers, and the timestamp at the bottom right corner is in minutes. So, as I start the movie, you can see that the movie frames are sped up 500 times normal speed. The first thing to note is that during development in this young rat pup, neurons are incredibly dynamic, extending and retracting protrusions called dendritic spines and filopodia on timescales of minutes. The movie will now repeat and zoom in on a dendritic filopodia extending and then contacting an axon at time point 90. As I replay the movie, you may also notice the wobbling of the dendrite branches as they move in response to the capillaries, as the shadows of the blood vessels expand and contract.
So by imaging the motility of these dendritic spines and measuring their movements of these synaptic structures, we can then correlate how this motility changes in response to changes in the sensory experience of the animal. If we deprive all of the whiskers at specific time points during the development of the animal, for example, for 2 days, starting at postnatal age 8, for example, 10, 12, 14, and 16, what you'll see on the y-axis is the motility of these dendritic spines in the deprived condition divided by the motility in the control condition, which is roughly 1 at postnatal age 10. And then you can see a sharp decrease of the motility in the deprived case, normalized by the control condition, at a specific time window, postnatal age 12, that then starts to increase again as the animal ages, which shows a critical period for this dendritic spine motility that coincides with the receptive field critical period. So to conclude these series of experiments investigating experience-dependent plasticity within the rodent barrel cortex, the development of the whisker map coincides with the receptive field formation, dendritic spine morphological dynamics or their motility, synaptogenesis, as well as the onset of the whisking behavior of the rodents. The sensory experience that refines these neural circuit wiring only refines it at a narrow critical period within the animal's life of about 2 days. So what have we learned from these studies about the development of cortical circuits? Formation of neural circuits is a sequential and multi-step process that encompasses several brain areas. Neural circuits for complex information processing develop later than simpler ones, as more complicated neurons integrate the information from the upstream, simpler neurons. Critical periods exist for certain developing brain circuits that absolutely require proper sensory experience for circuit refinement. And circuits that develop earlier in life are influenced less by sensory experience. Now I'd like to turn our attention towards adult experience-dependent plasticity. In other words, can an old dog learn new tricks? What is changing within the brain, and how are the neurons within the brain of an animal, even such as ourselves, changing when confronted with a novel sensory experience? If we look back at the classic 1960s experiments from Hubel and Wiesel, again, we could see in their measurements in the normal condition of a cat, the broad distribution of neurons within the visual cortex for responses to one eye versus the other, or to both eyes and everything in between. On the other hand, if we close one of the eyes for a period of a week or several weeks, then we could see that the majority of neurons now only respond to the spared eye. And this is shown again here in this summary, where we have the birth of the animal and the opening and close of the critical period. If we monocularly deprive the animal for a period of, for example, 1 week, again, we could see that the responses shift to the spared or non-deprived eye compared to the broad distribution in the normal cat. As I mentioned very briefly earlier, if we can deprive the animal of light, for example, using binocular deprivation or light deprivation, we can extend the normal critical period all the way up to this point, where monocular deprivation, which normally would have no effect in the adult, now actually does have an effect after 3 months of monocular deprivation. Is this actually the case for normal adult sensory experience? It seems that the close of the critical period may not be so absolute, but more relative, as follow-up experiments published in the 1990s showed using the adult rodent visual cortex. So here again, the dashed line here shows the close of the rodent visual cortex critical period, and monocular deprivation in the adult case actually causes a slight shift in the field-evoked visual potentials compared to the non-deprived eye in the control case. Much of our understanding of whether and how adult experience-dependent synaptic plasticity occurs within neurons in the brain was shown by the pioneering work of Michael Merzenich in the 1990s. And I've chosen a few examples of these experiments to illustrate his use of the adult owl monkey, and after training or changing the sensory experience of their fingers would cause changes in the receptive field maps in the primary somatosensory cortex of these monkeys. In this first series of experiments, Dr. Merzenich and colleagues trained monkeys to touch a rotating disc that rotated at 1 revolution per second and had alternating wedges of different depths. The difference in depths were 350 micrometers, or 350 thousandths of a millimeter, so a very small difference in depth. The monkeys were allowed to freely touch this rotating disc, and if they touched it with only the second and third digits, that is the index finger and the middle finger, with their tips of their finger at a certain pressure, the camera would detect this and the monkeys would be rewarded with a banana pellet. The monkeys were allowed to do this freely throughout the day, but most training sessions added up to about 2 hours per day. Then they recorded in primary somatosensory cortex of the adult owl monkey to see what happened. First, I will go over the topographic organization of the primate somatosensory cortex. Here we're looking at the brain of a monkey, and the right hemisphere is shown here with the front of the monkey facing this way, the back would be this way, and here we could see primary somatosensory cortex with the foot, leg, trunk, arm, hand, and face area shown here with area 3B shown here, and the hand region, if we take it, zoom in and rotate it this way, is shown here with rostral being up, or the front of the animal being up, facing this way, and the lateral side facing to the right here. If we were to stimulate each one of these areas on the left hand of the monkey, we have digit 1, digit 2, digit 3, 4, and 5. We can then find out and map these areas within the primate somatosensory cortex. As we can see here, digit 5, 4, 3, 2, and 1 are shown with very specific areas here, and the distal or ends of the fingertips have large representations here, 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 and here. And then as we move down caudally in the brain, we have the medial areas M here, or the middle parts of the fingers being represented here, and P for proximal, which is the areas closer to the palm of the hand until we reach P or palm. These shaded areas here correspond to the other side of the hand and will not be relevant for the following series of experiments. So what happens to the response properties of these neurons within primary somatosensory cortex as these monkeys are allowed to touch this spinning rotating disc with their second and third fingers for periods of several months? Again, we have the map of the hand in primary somatosensory cortex, and here we could see the representations of fingers number 2 and 3 and their sharp borders between the digits here, 2 and 3. After the rotation hits 2 and 3 very fast, and this constant stimulation for periods of a month, the first interesting result to note is an increase in the area represented by the distal part of finger number 2. The second interesting thing to note is the increase in the distal part, or neurons that respond to the distal regions of fingers 2 and 3. In similar experiments, they examined how the timing or near simultaneous activation of these fingers, or the inputs, for fingers 3 and 4 changed representations within the adult somatosensory cortex. And what they did was to induce artificial syndactyly. What that is by sewing up the skin between digits 3 and 4 and allowed the monkey to experience this for about 6 months. Syndactyly is a naturally occurring phenomenon where the skin between the fingers does not separate, and this is easily solved at birth by simply trimming up the skin so that the different fingers can separate. In the artificial syndactyly case, after 6 months, they found that this abolished the separation of the receptive field borders between digits 3 and 4. So here, as you can see in the results, the shaded areas represent neurons that had double-digit representations, which normally never occur in the adult monkey. That is, that these neurons here were responsive to stimulation of either one of these fingers, 3 or 4. So again, if we walk along here, stimulation from rostral to caudal, we could see responded normally, what normally would have been responsive to just a single digit, either digit 4 or either digit 3. These separate responses were abolished, and now stimulation of either 3 or 4 activated any of these neurons along here, whereas the somatotopic or topographic representation going from digit 5, 4, 3, 2, and 1 was preserved, except for along here. Again, you could see these neurons were responsive only to digit 5, this to digit 4, but as you got closer to the border to 3 and 4, what normally would have been the border between 3 and 4, these neurons are now responsive to both digits. To control for any changes that might have occurred due to the surgery, they simply clipped off the sutures and immediately recorded between the areas that were normally covered by the sutures between digits 3 and 4. And as you can see here, for A, B, and C, these also produced double-digit responses. Finally, in a series of very elegant experiments, they wanted to know whether or not by simply training the monkey or changing the behavior of the monkey, they could see similar changes in the receptive field maps within area 3B. The way they did this was by simply rewarding the monkey with a target signal of a raised bar, shown here, where the change in depth was only 50 to 100 microns. And this bar was only detected by digits 2, 3, and 4. And so what they found was that by simply training the monkey to detect this very subtle difference with digits 2, 3, and 4, they had abolished the fine separation between the digits 2, 3, and 4 and generated more response areas of these neurons for multiple digits. In these cases, 3-digit receptive fields, 2-digit receptive fields, and many more neurons were responsive to the distal regions of the fingers rather than the proximal. So what have we learned from these experiments about the plasticity of adult cortical circuits? Well, first, of course, is that lifelong learning can occur in the adult and that we can measure it. The level of plasticity, or the response magnitude, can also be quantified, and changes in the sensory experience in the adult can cause large-scale changes in the cortex. The receptive field map redistribution that occurs in the cortex at the expense of less used territory, so that more neurons are responsive to those neurons, to those inputs that are more actively used, and the timing of these inputs is important for this plasticity. So now we will move back to the rodent barrel cortex to examine some of the anatomical changes that occur in response to changes in the sensory experience of the adult animal. Again, as a reminder, the rodent barrel cortex is an excellent system to examine experience-dependent plasticity because of its one-to-one correspondence between whisker to barrel in a very precise topographic or somatotopic organization. And we know exactly where to look for any of these changes in experience-dependent synaptic plasticity that may occur in the cortex because of this one-to-one correspondence. By simply clipping a whisker, we could change the experience of the animal, and we know which barrel this corresponds to. One of the first experiments that examined adult plasticity within the barrel cortex was performed by Matt Diamond in 1993 and 1994. And what they did was to spare the D2 and D3 whiskers in a kind of rodent artificial syndactyly, which forced the animal to live for just 24 hours of whisker pairing. In these experiments, all of these recordings were performed only within the D2 barrel, shown here. So let's take a look at the control condition. This is separated within layers 2-3, layers 4, and layers 5-6 within the cortex, and the control condition is on the left-hand side here with the y-axis for these histograms as spikes evoked in 50 trials. Again, we could see in the control case the typical center-surround organization where the highest number of spikes evoked was through the principal whisker, or the D2 whisker. So stimulation of the D2 whisker evoked 40 spikes in 50 trials here, in layers 2-3, similarly in layer 4, and again in layer 5-6, whereas stimulation of the first-order surround whiskers, D1 and D3, evoked less of a response, and again in a decremental fashion to the second-order surround whiskers in the control case. After just 24 hours of pairing the D2 and the D3 whisker, again recording only from the D2 barrel, so these neurons shown here, you can see that the response to the paired whisker, in this case the D3 whisker, was increased, but only within this layers 2-3 and layers 5-6. Layer 4 was spared. Now to look at the anatomical manifestations or the structural changes of these neurons within the barrel cortex after changing the sensory experience of the adult, again we could turn to using 2-photon in vivo imaging, where we can image dendritic spines, dendrites, axons deep in the brain of the living animal. So the first thing to note is that dendritic spines are stable over timescales of hours in the adult barrel cortex. So here on the left-hand side is an image, a difference image of a rat at postnatal day 14, and you can see here that the dendritic spines are color-coded with green for new spines that occurred over a timescale of a difference between 1.5 hours, and red are spines that have disappeared over 1.5 hours. If we look at the adult case, in this mouse, we can see that all of the spines are incredibly stable over a timescale of 1.5 hours with no addition or retractions of spines. Therefore, we had to develop a technique that would allow for long-term chronic imaging of neurons over timescales of days, weeks, and even months. The way we did this was by using the unique vasculature, or vascular patterns of the brain, as well as the unique dendritic branching patterns of the cell to locate the same neuron over each day. This entailed creating a cranial window into the brain at around 8 weeks of age, then letting the animal recover for about 1 week, and then imaging daily for timescales of weeks or months. So this series of time-lapse images shows the timestamp here being 1 day, daily imaging for 8 days on this piece of dendrite shown here. When we imaged these dendritic branches over several weeks, we found that they were incredibly stable. Analysis of over 170 branches found 0 branch additions and eliminations over periods of months, with similar results for axons. Dendritic spines, however, were actually quite dynamic over timescales of days, and this movie shows a loop of 1 to 8 days, with each frame being 1 day. And first you'll notice these very large spines that are quite stable, whereas there are also dendritic spines that are quite dynamic that come and go over timescales of days, as well as new stable spines. We can quantify these dynamics in terms of, for example, turnover rate over time interval, for example, turnover rate per day, or as lifetimes, how long a spine lasted over a given time interval, for example, how long it lasted over a period of 4 days or 8 days. These yellow arrows, for example, indicate the presence of a spine, and the red arrows indicate the disappearance or retraction of a spine. In general, we found that about 20% of spines are gained and lost each day, and about half of spines survive for periods of more than a week. We also found that the creation of a new synapse or synaptic contact was due to the growth of a spine to a pre-existing axon. So, for example, if we take a look at this piece of dendrite here, with this dendritic branch shown here and an axon in the background highlighted by these yellow arrows, we could see the sampling of this axon here by the growth of these spines here, as well as a new spine that's shown here on day 8. Now, the evidence that the growth and retraction of a dendritic spine was the actual creation and elimination of a synapse was verified using a technique called serial section electron microscopy. So, for example, we have the in vivo 2-photon image of a dendrite coming from this pyramidal neuron, and we can visualize using this counter stain the unique branching pattern of the previously imaged dendrite after several days of imaging in vivo, and then using electron microscopy we can image the fine structure of this dendrite and the dendritic spines.
This movie, provided by Graham Knott and Egbert Welker, shows the serial reconstruction of electron microscopy images of a dendrite previously imaged in vivo for 8 consecutive days using 2-photon laser scanning microscopy. Blue regions indicate excitatory synapses, and red regions indicate inhibitory synapses.
So here's an example of a previously imaged dendrite from day 7 and day 8 from the in vivo image, and the reconstruction using serial section electron microscopy with the excitatory synapses labeled in green and the inhibitory synapses labeled in red, and these 2 new dendritic spines that occurred between day 7 and day 8 highlighted here by these yellow squares. When we zoom in on these newly formed dendritic spines, we can see they have the appropriate presynaptic density with synaptic vesicles and the postsynaptic thickening. Thus, the turnover of dendritic spines represents the actual change in synaptic connections in the brain of a living animal. We can quantify these synaptic dynamics, for example, by looking at their lifetimes. When we examined their synaptic lifetimes, we found that they were broadly distributed. For example, here on the x-axis we have their lifetime in days, and on the y-axis we have the percent or fraction of the total dendritic spines or synapses here. We found they fell into 3 broad categories or classes. One category, which was termed transient or unstable spines, lasted for a day or less. A second classification were called semi-stable spines with a single exponential distribution and an average lifetime or time constant of about 2 days. And a third class of spines which we called stable spines, which lasted for 8 days or longer. So here are some examples of a stable spine, for example. The yellow arrowhead points to a spine that lasted the entire imaging session. A semi-stable spine is this red one here that appeared on day 3 and then eventually disappeared at day 5. And then a transient spine that only appeared on day 5 and disappeared subsequently after. If we follow up and examine even the stable spines, we find that they also eventually turn over. So, for example, this spine here was imaged all the way for 8 days, but then after a month eventually disappeared, and these had a mean lifetime of about 110 days. So for a very quick summary, by imaging the dendritic dynamics in the brains of the living animal over timescales of weeks, we find that dendritic and axonal branches are relatively stable in the adult, synapse formation and elimination occur daily in the adult cortex, and that these synaptic lifetimes are broadly distributed. What does this have to do with experience-dependent plasticity? Well, again, we can turn to electrophysiological recordings in the barrel cortex to see how the response properties of these neurons change after changes in the sensory experience of the adult animal. So again, here we're looking at a recording from a neuron within the barrel cortex, and we have a response here to stimulation of the principal whisker. Here's the whisker stimulus event, and again, in each one of these red traces is an individual response, several tens of millivolts, for example, and the black trace shows the average response. They obviously have a stereotypical center surround organization, which is again shown here for stimulation of the first order surround whisker, and here is the average trace shown here. The principal whisker is an anatomical definition, so by adding a chemical to the electrode that then fills the neuron, after the experiment is over, we can then simply dissect the brain, take sections, and find the neuron again within this section after staining for the barrels, identify which barrel it is, and then identify what was truly the principal whisker and the first order and second order surround whiskers. To induce the largest amount of plasticity or competition between these barrels, the experimental paradigm that we chose is the chessboard deprivation. That is, by clipping every other whisker, a single whisker or a single barrel would always be surrounded by at least 3 other opposing conditions. So for example, this barrel here would be a spared whisker surrounded by 3 or 4 other deprived whiskers, and this deprived whisker or deprived barrel would be surrounded by 4 spared barrels. So again, in the control case, where all the whiskers are spared, we have stimulation in the stereotypical center surround organization, with the principal whisker generating the strongest response, with the first order whisker generating a slightly decreased response. After 3 days of chessboard deprivation, here we can clearly see that what normally anatomically defined would have been the principal whisker stimulation generates this response here, and stimulation of the first order spared whisker generates an even larger response. So in this case here, this neuron, principal whisker, is deprived for 3 days, and it no longer responds best to that whisker, and it responds better to a spared input. And this is summarized here, where again, in the control condition, we have stimulation of the principal whisker, first order surround or second order surround whisker causes a decreasing response, here normalized from 0 to 1 from with a center surround organization. And then after chessboard deprivation for 3 days, we can see that first the principal whisker or neurons within a barrel where the principal whisker is deprived, the first order surrounds are by definition a spared whisker, and we have a significant increase in the response to the spared whisker, whereas those neurons that had the principal whisker spared, we do not see much of a decrease in the response to the deprived whisker here. What are the structural correlates for this receptive field plasticity in the adult? Well, because we can image a neuron at high resolution over timescales longer than a week, we can directly observe these changes in synaptic connections as they occur in the animal when it has a novel sensory experience. So first, again, we look at the control case where we image every day for 4 days with all the whiskers spared, and we can measure the fraction or population of spines as they fall into these different classes or categories, with transient spines that last for a day or less, semi-stable spines, and stable spines. Once we induce a chessboard deprivation, clipping every other whisker, for 4 days we can image in the same animal again, and then measure the fraction of these spines that fall into the different categories. We find that the fraction of spines that fall into the stable population decreases significantly, with a concurrent increase in the fraction of spines that are the transient population. If we image neurons for the same 8 days, but these neurons are outside of barrel cortex, even after chessboard deprivation, again, we see no change in the fraction of spines that make up the different classes. If we look at the spine lifetime in terms of the turnover ratio, or in other words, the fraction of spines that are gained or lost over time, we could see, again, for the first 4 days in the control condition with all the whiskers spared, we see a normal turnover rate. And then after chessboard deprivation, In day 5 and 6, we see an increase in the turnover ratio, that is a destabilization of those stable spines and an increase in the turnover rate, or the increase in the population of the transient spines. Whereas outside of the barrel cortex, this is not the case. And this concurrent increase in the population of transient spines with a decrease in the population of stable spines results in a stable synaptic density. These results were followed up in this vivo lab by demonstrating that this novel sensory experience, or the chessboard deprivation pattern, eventually will stabilize these new synapses that are formed so that the turnover ratio will eventually level out. So again, we have a chessboard deprivation, this time for a period of almost 1 month. And again, we could see that the synapse density, or the spine density, remains the same throughout the entire protocol. If we look at the survival fraction, the chessboard whisker trimming here causes a slight decrease compared to control of the fraction of spines that survive. In other words, this increase in the turnover ratio is temporary, and eventually the survival fraction levels out. This is because, as you can see here, the fraction of new spines that occur that become then persistent for the rest of the imaging duration increases. So it seems that these transient spines that are constantly turning over are used as a pool so that during novel sensory experience, they can then refine that circuit, and those that are the most appropriate synapses to maintain are stabilized eventually. So to conclude these experiments in the adult rodent barrel cortex, we could see that receptive field plasticity can occur quite rapidly within 24 hours. This receptive field plasticity occurs in the pyramidal layers with no change observed in layer 4. The responses of neurons in the spared barrel to stimulation of the paired whisker increases, whereas in the chessboard deprivation protocol, we could see that neurons within the deprived barrel increase their responses to the spared whisker. Dendritic and axonal branches are stable within the adult brain, and dendritic spines and their growth and retraction are associated with synapse formation and elimination, and it's this day-to-day formation and elimination of synapses that is responsible for experience-dependent plasticity or refinement of neural circuits in the adult brain. I hope you found my lectures informative, and if you're interested in any of the topics that I've discussed, I encourage you to read about the details in the primary publications. Thank you very much for your attention.